such a full audience. I think Katie indicated this is the full house. It topped Paula's talk last month, so well done, guys. Well done, Tim. I've suggested Tim can add a little bit of jargon to his talk nowadays, and perhaps we can increase the number next time, right? Yeah. Anyway, tēnā koutou katoa, good afternoon, and a very warm welcome to Lincoln University. Thank you for coming this evening. Co-Professor Chad Hewitt, toko ingoa. I'm the provost here at Lincoln University, and it's my pleasure to be the host today. Our Excellent Series is one of our premier events. It celebrates the academic leadership and applied research excellence that defines Lincoln University. These events are an opportunity to highlight the work of scholars whose research contributes to a more sustainable, productive, and resilient Aotearoa New Zealand, and whose influence often extends across the globe. Today, we're proud to spotlight the work of Professor Tim Clough. Now, usually the Excellent Series is for a new professor. It's taken us 15 years, but we are celebrating Tim's excellence tonight. He is an internationally recognized expert in soil nitrogen dynamics and climate-smart agriculture. Professor Clough's research focuses on how nitrogen moves and transforms in pastoral systems, especially following urine deposition and fertilizer use. His pioneering work has significantly advanced our understanding of nitrous oxide emissions, potent greenhouse gas, and how to reduce them. By combining field experiments with sophisticated tools such as isotopic tracing, Professor Clough has helped shape both policy and practice, from informing national greenhouse gas inventories to developing practical nutrient management strategies that support more sustainable farming. More recently, he's extended his nitrogen expertise to aquatic systems, exploring nitrate removal and emissions in the Vedos zone and freshwater environments. His work exemplifies the kind of integrated, solution-focused science that's essential for addressing climate and environmental challenges in agriculture. Professor Clough is a fellow of both the New Zealand Soil Science Society and the Soil Science Society of America, a testament to the impact and reach of his work. Join me in welcoming Professor Tim Clough.
All right, thanks, Chad, for that. Sound okay there? Yep. Okay, so I was wondering, what am I going to talk about? So I put a broad topic up there, and at some point we'll touch on the implications, the environment and the climate, but we want to talk about forms of reactive N, what they are, where they're coming from, where they're going to. So nitrogen was discovered sort of in the late 1700s as an essential nutrient, and it predominantly exists in the air. 78% of the air we're breathing is dinitrogen, and it's non-reactive, and it's comprised of two nitrogen atoms with this triple bond, which makes it very strong, and it's very hard to break that bond. Reactive nitrogen, on the other hand, is all forms of nitrogen that are present in the environment, except for that dinitrogen. And some of the names have probably come across: ammonia. If you go home and you open up the handy Andy bottle and take the top off and have a whiff, that's ammonia that you smell. Nitrogen oxides, NOx. If you were coming off the Bombays into Auckland, there's the brown haze. That's probably nitrogen dioxide, NO2. But it's also being generated in our agricultural systems. Nitrous oxide is our greenhouse gas, or one of them, one of the three major ones. And nitrate you would have heard about in terms of nitrate leaching. We've also got these organic forms of reactive N, urea, as in urea fertiliser, amines, proteins, and many other forms. So this list is by no means exhaustive. So before humans came along, that non-reactive nitrogen in the atmosphere was able to get down to ground level and could be used by microbes. And natural creation of reactive N occurs through lightning strikes. So there's enough energy there in the lightning to combine oxygen, nitrogen to make nitric oxide, NO. And that's oxidized pretty quickly to nitrogen dioxide and then to nitric acid. And then that's precipitated out in rainfall or dry deposition. And that's how it gets into the environment for microbes or plants, organisms to be able to use. Wildfires by combustion are also creating reactive nitrogen. So there are a couple of natural ways. And the third way is biological nitrogen fixation. So this was discovered towards the end of the 19th century. And there may be free living organisms that can do this, that can pull that out of the atmosphere down into their bodies, and that can then cycle through the soil. Or one that we're more familiar with is a symbiotic relationship with a plant. Often we're referring to these as legumes, and we'll have an organism like rhizobia, which can pull that out of the atmosphere, give the plant some nitrogen, ammonium, and in exchange, those organisms getting something like carbon supply. So there's a symbiotic relationship going on. And in natural ecosystems, there are many species that are doing that. Trees, there can be aquatic systems, and terrestrial systems where BNF is occurring. So we talk about natural BNF, and we talk about anthropogenic BNF. If we knock over a forest and put in soybeans or something, a legume, we've set aside the natural ecosystem and we've now put in more agricultural BNF, biological nitrogen fixation. So at the end of the 19th century, this guy, Sir William Crookes, who's president of the British Association for the Advancement of Science, noted that all England and all civilised nations stand in deadly peril of not having enough to eat. And this was due to increasing food demands and a lack of nitrogen. So this sent them running around civilised nations looking to mine a source of nitrogen. So that was guano, bird deposits, or some of the salt deposits in South America. But then just after this, 1913, Haber and Bosch, who were subsequently awarded Nobel Prizes, and you can ask if that was a good thing or not later on, they developed and perfected the industrial method for generating ammonia by taking N2 out of the atmosphere. And to do that, you have to combine that with hydrogen. And at the moment, about 1% of the our global production of methane goes into this production of ammonia. And that can be then used readily to make fertilisers or explosives. And an example of this making fertiliser is the Kapuni plant, which is running out of gas, up in the North Island there. So this is now the focus and the way that the global population gets cheap nitrogen. And it, as I said, goes on to be used for many uses. The efficiency of Haber-Bosch nitrogen, and I was quite staggered when I read this, is apparently if we've got 100 units of fertiliser derived from Haber-Bosch, about 14 units will end up being consumed by vegetarian, about 4 units by carnivore. And I'm willing to believe this is right, because this comes from James Galloway. He's a global authority on determining such budgets. So that was a surprising thing to read in some ways. The net result of this relatively cheap nitrogen fertiliser has been an explosion in the global population. We're currently sitting, I think, at about 8.2 million. So this graph is about 10 years out of date. But the point to make is that 1 in 2 people, on average, now depend on synthetic fertiliser nitrogen for their diet. So a human being needs something like 2 kilograms of nitrogen per year, I understand. And on average, we make much more than that, 27 grams, I think. So here's our reactive nitrogen inputs on a global scale. A teragram is a megaton or a million tons. And we've got our natural BNF coming in there, and the green arrows coming down, lightning BNF, but we've also got BNF occurring in the oceans as well. There's fixation going on there. And we've got our anthropogenic creation of reactive nitrogen. Combustion, any oil or coal that's being combusted contains nitrogen that's releasing things like nitrous oxide. Our fertiliser production we just mentioned, and we've mentioned agricultural BNF. So soybean production in particular, but also legumes used in forages or seed crops would also come into that category. And in 1965, our anthropogenic creation of that reactive nitrogen exceeds the natural production. So for the last 45, 50 years, we've been producing more than the natural environment. So if reactive nitrogen doesn't make it to the plate, where is it going? It can be stored within the system. So in an ag system, maybe it's stored in the soil, on the soil surfaces somewhere there, immobilised by microbes. It could be transferred to another system. So an example would be nitrate leaching out of the soil, moving off to an aquatic system, the oceans perhaps. Or if we're lucky, it's been denitrified back to N2, the inert compound nitrogen. So what is denitrification and how do we get N2? So it's a microbial process, and it's taking, in this case, nitrate to nitrite to nitric oxide to nitrous oxide, our greenhouse gas, to dinitrogen. If we're lucky, this will go all the way to completion and make the inert N2 gas. But quite often we get nitrous oxide leaking out of the system, whereas we rarely see nitrite or nitric oxide. They're quite reactive. But this process is anaerobic. It requires carbon substrate. These are heterotrophs, and it's sensitive to anything that microbes would be sensitive to. Each step is driven by an enzyme, so we can upset enzymes by changing the pH or the temperature. So it's not something you can just turn on, but you can manipulate this pathway. And an example of that would be wastewater treatment plants, where they try and manipulate this to get rid of nitrogen. So how do we follow reactive nitrogen in the environment? So I'm an addict for using stable isotopes. And the stable isotopes of nitrogen that we would use would be this one called 15N. If we went out there and sampled some grass, or we took a blood sample out of ourselves, put it through the mass heat, we would find that about 0.4% of our nitrogen atoms are this heavy nitrogen atom, the 15N. The remainder are the lighter one, the nitrogen 14N. And we can look at the ratio of these to determine and follow reactive nitrogen. And the way we would follow a compound would be to dose that compound and then put it in the environment, if we're going to use enriched tracing, which is, I think, one of the easier ways of doing it. So an example might be, you want to know what's happening to urea fertiliser. You can add 15N urea to that fertiliser when you apply it to the crop that you're interested in studying. Or if you want to know what's happening to nitrate as it leaches through the environment, you could actually put labelled nitrate into the environment to follow the fate of that nitrate. And isotopes don't lie. You can screw up the measurements, but if you get the data out, you've got this very solid evidence for what has happened to the nitrogen. It's disappeared or it's formed a new product. So an example of such an experiment was this one here, where we looked at the recovery of dairy cattle urine, and we doctored that up with labelled urine because cattle urine is predominantly urea. About 80% of it is urea. And then here we were interested in looking at 4 soil types: a silt, a sandy loam, a peat, and a clay. And we've got on the y-axis there the percentage 15N recovery. So you can look at that 15N composition in each of the pools that you're looking at. And here we've got the plants in green, soil in brown, leaching in the hatched one. And we didn't measure any gases here. So this is our apparent gaseous loss. But if we were trying to look at storage, that would be in the soil or the plants. The plant storage is temporary, so it's going to be grazed and turned over again. The leachate here is being the transfer, and the gaseous emissions could also be a transfer, so being transferred into the atmosphere. And it's worth noting there maybe that of the N that's gone on, roughly 50% has stayed in the system. So we're getting losses from the system. If we think about our ag systems, I use that term broadly. I mean, horticulture, as I understand it, citrus is a heavy user of fertiliser, kiwi fruit, vegetable production. So, but using the term broadly in agriculture, our key sources of our key N inputs contributing to reactive N creation are fertiliser, particularly our legumes, high in N. While they're growing, they're not contributing too much, but if they're going to go through a grazing animal, then we end up with one of our key sources of N, and that's the urine that's excreted, or the dung. So ruminants, they'll consume, ingest their fodder. The amount of N going out in the dung is relatively stable, but as that feed becomes more nitrogen rich, they'll excrete more through the urine stream. So these are our key sources that we need to consider. If we look first at some of these forms of reactive N that we're losing, and we'll look at ammonia here, the dominant form coming in from excreta or fertiliser is urea. So in New Zealand, the use of urea fertiliser has gone up about 600% since about 1990, driven largely by the dairy industry, as I understand it. Urea is broken down by an enzyme called urease that's ubiquitous, it's everywhere, and it forms ammonium and this thing here, OH minus hydroxyl. If you remember from your chemistry, this drives up the pH, and if we've got a high pH in ammonium, we'll get ammonia formed. So this is our reactive N that can potentially move into the atmosphere. And the amount of ammonia being lost is quite significant. If we look at the greenhouse gas inventory for New Zealand, on average, we're taking a value of 10% of ruminant urine excreta is going off as ammonia. And I think it's about 0.67% for urea fertiliser at the moment. So these, at 10%, it's starting to become a bit of an economic loss. And that's for excreta. Fertiliser ammonia loss, sorry, I gave the wrong number there. Fertiliser loss for ammonia can range from 1 to about 50%, and it depends on your environmental conditions. If you've got dry soil, lots of wind, short canopy cover, you'll have high emissions of ammonia. So farmers are told to put urea on before irrigation to try and water it in, or put it on before rainfall to minimise the chance of ammonia loss from this urea fertiliser. So at about $1,000 a tonne, even 10% starts to become significant in terms of a loss. Here's an example of the change in pH that occurs under a urine patch. And this was Kate Lewin who did this a few years ago. We've got 100 by 100 centimetres, a metre squared. And you can see here on the periphery, our native pH, if you like, is 6 or 6.5. And after 24 hours, the middle of that patch where the urine is concentrated has gone to about pH 9. So a 1 pH unit increase can increase ammonia volatilization about 10 times. So here that's increased about 2.5 times. So this is why urine patches are hotspots for ammonia emissions. And it's the same under a urea prill. The same process is going on. You'll find a high pH there. So why do we care about ammonia volatilisation? Ammonia goes up, reacts with nitrous or uric acid in the atmosphere, and you'll get acid rain. So that's not something we even think about here in New Zealand. And we generally don't think about many of these issues here in New Zealand, except maybe the economic one, because we think about ammonia blowing offshore and it's gone outside our country's boundary, end of story. But if you're in Europe or North America maybe, it was a different story. So back in the 70s onwards, acid rain was quite a problem and ammonia was contributing to that. So there was quite an effort there to reduce ammonia emissions and think about different ways of applying nitrogen and slurries there. If we've got ammonia going to the atmosphere, it's coming down, it's acting like a fertiliser. And some other organisms there, like those that can oxidise methane in the soil, can also use N that's deposited onto the soil surface. And this ammonia goes to ammonium, and that's what these organisms use. So instead of doing the good job of chewing up methane and oxidising that, they'll switch to using ammonium because it's more energy efficient. If that ammonia is blowing downstream and landing, it's acting as a fertiliser. It's what we call an indirect source for nitrous oxide emissions. So it's an indirect source for N2O as well. And that gets taken into account in our greenhouse gas inventory. And again, depending where you are and how much N is coming in, if you're fertilising the system downwind, you'll potentially change the biodiversity in there. And I'll show you a picture in a moment of that from Minnesota. And I mentioned the economic loss. So apologies, this is a bit hard to see, but I thought it was a good graphical demonstration of how ammonia deposition changes the composition of species. So these were native grasslands in Minnesota. Apparently there's 20 or 30 species there. And in this work by Vertusik, who did a lot of work early on, what you're left with after a period of time is one or two species dominated by exotics. And that can be happening in terrestrial systems, maybe also aquatic systems. If we look at a global map, so this is a recent one, 2025, how much N are we depositing? Well, globally in some areas, maybe it's about 50 kilograms of N, and the hotspots are in Asia. Europe's cleaned up a lot of what's going on there. Maybe about 20 in some places. And I thought it was kind of good. New Zealand's off the map here. We have no data. You can go back and find paper by 2006 or Jakarta, where Louis Schipper and Troy based, and they tried to work out what's going on. It's just not something we measure because policymakers don't want to know about it because they don't want more nitrogen coming into the system. It's going offshore. This is very handy. And it's not really a productive issue or a production issue because the rates coming in are quite low. So I think from Parfitt's 2006 work, it's about 7, but less than 10 kgs of N per hectare, depending where in New Zealand you are. And at Bering Head, where they measure gases, NIWA, they find quite low concentrations of ammonia, but a lot of that air is not coming across New Zealand's landmass. So we don't really know what's happening. So what about nitrate? So nitrate leaches with drainage from the soil. It's got a negative charge. It can't hang on to those cation exchange sites in the soil. So if we've got ammonia volatilization, that's happened. We're left with NH4 plus in the soil. And ammonia volatilization should be all over in about a week. We'll be left with NH4 plus. This can be taken up by the plant or undergo this process here called nitrification. The end result is nitrate. Now, plants can also take that up, and it depends what the plant type is and what its preference is. But if the plant doesn't take up that ammonium, we'll get nitrate present. And usually within about 2 weeks, that ammonium's been consumed or it's all moved through to nitrate, and we've got a nitrate pool sitting in the soil. So this was work from Laura Buckthorpe's PhD in the Waikato, who was looking at the overlap of fertiliser and urine patches. And the hypothesis was that there'd be more leaching, more NTO emissions where fertiliser and urine overlapped. And what we found was that none of the fertiliser came through in the leachate. We labelled the fertiliser up with the isotope. And she was also looking at autumn versus spring leaching of urine deposition beds. So not surprisingly, most of the nitrate leached here came from the autumn deposition. And there was very little leaching following the spring deposition of urine. And the amount of fertiliser going on didn't change that. So 48% of that urine applied was being lost as nitrate. And so that was done in lysimeters, which measure gases and leachates and the fate in the soil at the same time. So that's an economic loss of the N out of your system. But what we did by chance was also measure the dissolved organic nitrogen, because we noticed a bit of brown colour in the leachates coming out. But what surprised us a lot was that the dissolved organic nitrogen, something we never measure, was about 1/3 the amount of urine coming out of the system. So again, we haven't gone back to this much at all, but I think it's a gap in our reactive nitrogen understanding. That's dissolved organic nitrogen that's come off the soil. What's replaced it? Is it some of the nitrogen that was in the urine or the fertiliser? Is that knocked the dissolved organic nitrogen off? We don't really know. And it's probably got something to do with that high pH that we saw under a urine patch. So scope for more work there. So nitrate is potentially an economic loss. We know it leads to eutrophication of waterways and oceans. And some of the stuff that's been in the media recently, there's a dead zone in the ocean off the South Australian coast. And one of the reasons that's formed is the runoff of nutrients from the land mass there. You've got phosphorus and nitrogen creating algal blooms, creating this area that's devoid of oxygen. And there's other areas around the world that are famous for it, like in the Gulf of Mexico. And this can happen in small ways, doesn't just happen in oceans. We're going to get indirect NTO emissions coming from some of these waterways where nitrate has moved into. Changes in biodiversity in your aquatic system, and then potentially health. I don't think the science is in on this. I've got papers there showing that the human body produces nitrate in the gut. So it's a very controversial area, but I think we still need more science to be sure. So another reactive form of nitrogen is our nitrous oxide. We've got direct and indirect emissions, which I've mentioned. This is a schematic from the IPCC guidelines. So we've got fertiliser, organic fertilisers, excreta, urine and dung, crop residues. They're being put onto a land mass somewhere to increase production. If the N2O comes off from where that application occurs, we call it a direct emission, that black arrow there. If we get ammonia volatilization and nitrate leaching, shifting that nitrogen around and it ends up somewhere else, it can emit the N2O at a site where the deposition didn't occur. So this is the indirect emission. So we get loss of N2O from nitrification, and we're still understanding the nitrification cycle. So in about 2015, the compound in the middle here, nitric oxide, NO, was only confirmed as being a member of this nitrification sequence. Before that, if you go back to some of the textbooks I had at university, there's lots of arrows and lots of question marks, really. But the net outcome is that some of these nitrifiers, the bacteria, carry denitrifying enzymes. So if the system goes hypoxic, they switch them on and they convert NO2 minus through to N2O. So that gives them energy until the system becomes aerobic again. But most of our losses as N2O come from this denitrification pathway we saw earlier. And as I mentioned there, it needs carbon, which we've got in plenty in terms of root exudates from plants. It needs a suitable pH, so it can't be too acidic. If it's too acidic, we'll only get nitrous oxide forming. We won't get to N2. Temperature, it slows down at sort of winter temperatures, 5 degrees. And so because it's anaerobic, it's dependent on the system going anaerobic. And the thing driving that most is soil water content in our systems. Potentially you'll get some anaerobic conditions if you increase respiration and consumption of oxygen, but primarily it's the water content. So how do we measure our N2O? This is from Uruguay with colleagues there. This is a SU alpha, I was going to say pasture, but it's just a massive area that they've planted. And we would start by putting a base into the ground like this. On the right-hand side here, it's got an annular channel filled with water. We put a chamber in, the water forms a gas-tight seal. And over time, we take gas samples to determine the increase in concentration in the headspace. We take those samples back to the GC and we generate some horrendous graphs like this, but we've got time along the bottom. We've got a rate of N2O flux on the y-axis. So if we look at the middle one, we've got the flux tracking along those black symbols there, and we can get a daily flux and see what's happening over time. The blue bars were rainfall events, and we'll often see spikes in the flux during the rainfall event. But what we can then do is work out the cumulative N2O flux over time. And again, this was Laura's PhD work, and we found that of the urine N, about 0.4 to 0.7% of the urine N applied came off as N2O. Now that would be very, very typical for grazed pasture systems in New Zealand. Adding the fertiliser on top of that didn't really exacerbate the N2O emissions. It was less than 0.04% of the 15 N applied. So within New Zealand, we've done ag research, plant food, land care. There have been many of these studies done, probably hundreds now. So the Intergovernmental Panel on Climate Change, the IPCC, initially said the default for N2O emissions was 2% of the N going into the system. So through the work that's been done in New Zealand, we've lowered that, and our default now sits below 1% for some topographies and types of farming operation. We can also examine these N2O fluxes from our indirect emissions. So this is the L2 stream a few kilometres away down here. We're trying to determine how much N2O is coming off our water bodies. The IPCC dictates that the N2O flux should be a fraction of the nitrate moving through the system. What we could show from this work is that our N2O flux is significantly lower by orders of magnitude than the nitrate moving through the system. So that and some other studies that have been done around the world have now pushed the IPCC to help lower their default emission factors for some of these indirect emissions. The bigger streams and rivers aren't necessarily the first port of call for that nitrate that's moving through the system. This is an agricultural drain off Ellesmere Road, a couple of k's away, and this was Manjula's master's project. And rather than put chambers here, because there's no room, we're measuring the dissolved N2O concentration and determining the transfer velocity of that gas into the atmosphere to predict the flux of the N2O. And there's still data sitting here to write up. The IPCC also talk about not just nitrate, but also ammonium, NH+, cycling in our waterways, and they attribute some N2O to that also being cycled. So in this experiment, we've also got labelled ammonium travelling through the system to look at the N2O emissions, which again were very, very low. So this is a table from the greenhouse gas inventory, which you can find on the MFE website. But for N2O, the big categories are the ones I've mentioned: synthetic fertilisers and excreta onto paddocks. And because they're linked, the indirect emissions from volatilisation and leaching also contribute. So of New Zealand's agricultural greenhouse gases, N2O is about 15–16% there at the moment. Why do we care about N2O? It's not really an economic factor, less than 1% maybe, but it's a global warming gas and it's got a 100-year lifetime in the atmosphere. And it's potent as a greenhouse gas. Currently, it's rated at being 273 times more potent than CO2 at trapping out infrared energy. And it's also got a secondary role, and at the moment, they assign to nitrous oxide this thing called an ozone depleting potential, ODP. And the way that N2O is removed from the atmosphere is during chemical breakdown in the atmosphere with wavelengths of light, and that generates NO, nitric oxide, is one of the products. That reacts with ozone, and there's more reactions there. The net result is that ozone is taken out of the stratosphere. So at the moment, we've got a Montreal Protocol globally signed up to in the 1980s. That's helped remove CFCs, which I think we're all familiar with, were potentially depleting the ozone. Ozone is recovering, but now we've still got nitrous oxide in the atmosphere, and that's increasing exponentially at the moment. So, and it's there for 100 years as well. So the more we can do to lower N2O emissions, the better. So we've got reactive nitrogen there, ammonia, nitrate, N2O are examples. There's a couple I haven't mentioned. We've got a visiting professor here from Japan, Chika. She's going to be looking at something called HONO, H-O-N-O, which I know very little about, but there are still other forms of reactive nitrogen that we need to look at. But what we've got here is a paper that was a call for more data on N2 fluxes. Modelers have been doing a lot of work with reactive nitrogen to understand where it's coming from, where it's going to, N2O, nitrate, ammonia, NOx. But we have very few data sets on N2 fluxes. So how do we understand that reactive nitrogen has left the system if we can't measure N2 or model and know when it's leaving the system? So in 1998, I got lucky and went to Northern Ireland to Belfast to work with two guys, not these two, but there were plenty of guns about. It's a very perturbing experience. But I worked with Jimmy and Ronnie, and they were at the height of their game in using 15N isotopes to study N2 fluxes. So they were really getting into all the equations, calculations, assumptions, and theory behind it. So it was a chance to learn a lot. And I've been able to apply that a bit over the years. And now this is a PhD student from Kieran Ding, who's with Ag Research. And Diana Selby also did some work in Northern Ireland looking at N2 fluxes as well. But I think these are some of the few data sets that we actually have on pasture systems and trying to work out how much N2 are we returning back up to the atmosphere. So Kieran's project looked at labelling up urine, putting it onto the soil, a fantastic number of replicates, N equals 6. So she worked like a Trojan, but produced this very nice data set. And we see high N2 fluxes occurring, much higher than the N2O. And the bottom graph here, at about day 38, 40, we see N2 fluxes starting to go up. And they shouldn't have been, because the soil was becoming aerobic. So denitrification is anaerobic, it's got to be waterlogged to be happening. And we measured the soil physics at the same time. That's why we knew the soil was aerobic. So what's happening is that the N2 is being produced while the soil is anaerobic, but it can't escape. Gas gets out of the soil by diffusing, mostly. And so it couldn't diffuse out of the soil until the water drained. And then we start to see this much larger spike of N2 flux. So that was interesting in all sorts of ways, but a lot of people use the ratio of N2O to N2 to try and predict N2 fluxes. And if we'd done that, we would have got quite a different answer and quite the wrong story about what was happening. So potentially we can now collect more N2 flux data from our agricultural systems. So I'll leave it there, and we've sort of only touched on this topic, and maybe given the interest we should be doing something a bit more detailed. So some of the key questions still to go are, where is our reactive N going and how much is going to N2? And understanding things like dissolved organic nitrogen in our ag systems. There's a few things still we know nothing about. So thank you.
What a fantastic talk. Now, before we go into questions, and I'm sure that there are going to be plenty, I just want to make sure, because I have a habit of forgetting to give the gift to the speaker. So thank you very much. That was a fantastic speech. Open the questions up to the audience.
Yeah, thanks, Tim, for interesting talk. My question is, how about reverse reaction? I mean, converting nitrate to ammonium. Some microbes probably do that, and they call it nitrate ammonium.
I mean, there's heaps I haven't talked about. So dissimilatory nitrate reduction to ammonia, DNRA, and under anaerobic conditions. But people talk about C carbon to nitrate ratios, but I think it's more about the redox of the system, which people generally don't measure. But if you could measure redox, you could probably find the particular switch when DNRA happens. But it's a way that microbes or ecosystems have got of conserving nitrogen. It doesn't leach out, it gets converted to ammonia. So one of the challenges, and something I didn't mention, we've got as an agricultural group, is keeping nitrogen as ammonium in the system. And so that's one way nature has got doing it. But we're looking at things like using nitrification inhibitors, either synthetic or the huge interest that's in biological nitrification inhibition. So plantain is one that's being looked at a lot, but there are other plants. So over the years, we've bred plants to be really good at growing well where we throw them nitrogen. And we've probably bred out any natural nitrification inhibition. And people are going back and looking at all the land-raised cultivars and things, and they're identifying nitrification inhibition in some of these old cultivars. So there's huge interest in trying to understand how to breed that in. And of course, GE is potentially going to make that possible.
I think you mentioned that the level of nitrous oxide in the atmosphere was increasing exponentially. I hope I haven't misquoted you there. What are the major sources in the atmosphere that are causing this to occur?
So the increase or the emissions of nitrous oxide, about half of them are anthropogenic. So there are large sources of naturally occurring N2O. So we've got N cycling out there in natural ecosystems, so N2O does come off those. But the biggest drivers are excreta and fertilisers. So some of the recent data can look at the atmospheric N2O and the isotopic composition on that, and from that they can infer the source, and you can see that it links to excreta and nitrogen fertilisers. And you can do the same with CO2 and look at the fossil fuel and isotopic composition. So anywhere we're using fertiliser, we've got this poor end use efficiency going on with fertiliser. Not so bad, I don't think, in our pasture systems. We're only putting on 25 kgs of N, plants doing well to take it up, but relatively it's still low compared to you know, where the rest is going. Yeah.
Great talk, thanks, Tim. New Zealand farmers have really had quite a big change in recent years because they now have a cap on nitrogen fertiliser use. And those intensive farmers might be using less than half the fertiliser that they were just 3 or 4 years ago. What impact do you think is that going to have on in losses in New Zealand?
From that actual 1 kg then going on, or 100 kgs, it's not going to change the loss from that fertiliser that goes on. But as a nation, we will see that tracking potentially into our greenhouse gas inventory. It's sensitive to things like drop in animal numbers due to drought and things like that. And if we see a decrease in synthetic fertiliser use, that will show up in the greenhouse gas inventory for sure. And then, you know, can we model, because we wouldn't have the data to perhaps go out and do it, but maybe we could model what would happen on a catchment scale and look at emissions such as nitrate leaching perhaps.
So what impact can the addition of urease inhibitors, particularly to urea fertiliser, have, and then potentially nitrification inhibitors applied to pastures?
Well, we've got some knowledge of it, and if we use a nitrification inhibitor, and I can find papers arguing both sides, it means that we've got more NH4 plus there. It's not going through to nitrate. But initially, if your form of fertiliser is urea, you've got a high pH and suddenly you're leaving more NH4 plus there. So you're potentially contributing more ammonia. So if you can use a urease inhibitor to slow down the rate of NH4 plus formation, then I think that's a good thing. But do we know enough about them, and is it economic for a farmer or a horticultural manager to use? That would be a question, I think. If you can slow down the loss of nitrogen from the system, so if you're just going to use a nitrification inhibitor, urease works, ammonium's there, then you're giving the plant more time to take up the nitrogen, and some data supports that very well, and you're definitely going to reduce your nitrate leaching events and your N2O emissions, and data supports that really well. So the question is how to get it used. Well, first identify the nitrification inhibitor, because it's synthetic or plant, and you know they're still in the pipeline, they're not on the market perhaps. But if you can get them out on the market, they will definitely have an impact. The question then becomes cost and farmer uptake.
And thanks for a great talk. Really interesting. One of the sources of nitrogen that was mentioned in one of your slides, but you didn't talk to a lot, was soil organic matter mineralization, right? Yeah. And we know that New Zealand soils tend to have relatively higher organic matter levels. We can have very high levels of nitrogen mineralization, and there are existing emission factors for predicting nitrous oxide emissions from mineralization. How confident do you think we can be in those emission factors, and how important do you think the mineralization might be as a source of increasing nitrous oxide emissions from our agricultural production systems?
Yeah, I don't know. I don't think I'd be confident. I mean, I don't know if we've got enough data, and that's one of the good things about trying to put some slides together. You think, oh, light bulb moment. Because I thought of you, Mike, with your carbon sequestration onto soil mineral surfaces, and then I was thinking, well, what do we know about nitrogen there? I mean, we can look at ratios of carbon and nitrogen and things like this, but that urine patch destroys the soil pH, and we know solubilization of soil organic matter happens. So how much are we stripping off the mineral surfaces, and what are the dynamics following that urine patch event? Yeah, but I think it's definitely an area of work.
You had a light bulb moment for an alternative to the harbour boss process. We've got another means other than BNF for capturing.
Yeah, well,
Only.
you know, I was wondering whether we'd ever taken a Nobel Prize. But yeah, I think we need to look at a different fertiliser. So we know if we've got an organic fertiliser, NTO emissions are low because normally in our system we have AOV bacteria that occupy that niche of enrich soil underneath fertiliser or urine patches. But if we put organic material in there, it's quite often archaea, apparently, microbiologists say, and they produce less N2O per unit of nitrogen processed. So in terms of N2O, we'll get lower N2O emissions. And if that's going to take more time to mineralise down, that gives the plants more time. So using this cheap urea fertiliser, it's just not ideal. I mean, that's where legumes are better. You still get some of these issues when you get the urine excreta. There's many other things we haven't talked about here, like if you can get the N content of your feed down lower, and maybe this is where Regen Ag goes, you've got less in the urine patch and these sorts of things, and that'll lower emissions, lower leaching and things as well. But I think if someone could come up with an organic fertiliser, I know there's some poultry organic fertilisers and things like that, it's definitely something that we need to look at, especially if Kapuni goes off stream. It becomes too expensive. Yeah.
New Zealand soils have relatively high levels of organic matter, hence relatively high levels of nitrogen in them.
Yeah.
Where did it all come from originally?
Well, it's going to have to come from biological activity, so it's organic matter, isn't it? And you say we got high levels. Talking to farmers south of Brisbane at Gatton the other week, fertiliser used to be a luxury for them, and they would get nitrogen coming into the crop from mineralisation of organic matter. Now they're saying their soils are so low in nitrogen from years and years of cropping, they have to use fertiliser to get a yield. So, you know, that organic matter component is really important to maintain.
Globally, production of rice and paddy fields, how much does that add to nitrous oxide emissions?
If you're doing flooded rice production, it's quite low.
Quite low.
Where's Chimindu, correct? Yep, but methane is the issue.
Right.
But if you're doing rice production where you've got periods of drainage and flooding and sort of cycling that water up and down, that will lead to more N2O emissions. But on a global basis, I don't know what the percentage from rice paddy production is.
I think, if I'm judging correctly, Tim probably is looking forward to a drink and some discussion. So please join me in thanking Tim for a fantastic talk. Join us outside for drinks and nibbles. Thank you.